Hello, I'm Bob Stebbin. I'm the Outreach Lead for the Wales Kidney Research Unit. The unit represents kidney researchers throughout Wales, and you're going to see a day in the life of the researchers here in Cardiff.
Okay, so I'm doing a reverse transcription. So that's to reverse transcribe or change my RNA that I've extracted from urine samples from patients into cDNA so that we can measure these things called microRNAs in their urine. The aim of that is to predict whether these microRNAs change to better predict whether these diabetic patients will go on to get diabetic kidney disease.
So the project that I'm doing is mainly looking at what we'll be able to apply to transplant kidneys. So we're looking at the injury that the kidneys get in that time when they don't have blood supply, and basically looking at ways that we can reduce that so we can improve the outcomes of kidney transplants. So I've got here kidney tissue, which we've basically dissolved in an agent called Trizol. And the plan for today is to extract the RNA from it, so basically the genetic material. Basically now I'm using the centrifuge to spin the liquid in the epidermis. So by the time it gets to the end of this programme, it will be split into its different components, and the RNA will be nicely sitting at the top, so then I can take that off, and then all the experiments from here on will just use that RNA and get rid of all the waste.
So currently I'm working on a research project that is looking at a specific receptor involved in fibrosis. So fibrosis is in effect scarring of soft tissues like organs such as liver, kidneys. The next stage then is to silence the receptor that I think is involved in fibrosis and see whether it reduces this contraction.
So my project is looking at the immune response of peritoneal dialysis patients, especially when they get a peritonitis infection. So I'm looking at specific biomarkers so we can identify the infection quickly, and then we can hopefully treat it more effectively. I'm currently in the final year of my PhD, so I'm writing up my project with the aim of publishing it soon, and hopefully going to move on to something similar.
I have been growing up some kidney cells in the lab, and I've been tinkering about with them and knocking out a gene that I think might be important in diabetic kidney disease, which is what I'm interested in. But this is purely just to see whether I've managed to capture the protein on this bit of membrane, because if I haven't got it there, there's not much point in staining it for anything else. Nope. Should we keep going until I find it?
Welcome to science.
This is science! There is actually some protein on this one, right at the bottom. There's like a little smear of darker. Can you see that? So this is the ladder. This bit here is the ladder that tells me how big my proteins are. And then the proteins appear and I can see which protein it is because I know what the molecular size of it based on that ladder is. And can you see there's like a one there, one there, one there? So that is a bit of protein developing. But that explains why I'm not getting very good results from these plots, so probably back to the drawing board for this one. Well, hopefully what I'm trying to do is look for a new marker of diabetic kidney disease, so this could be useful for therapeutics, or something that you can target to control the levels of that might improve your responses in diabetic kidney disease, or it could be something that we use as like a biomarker, so something that might be in patients' urine or blood, for example, that we could use to detect diabetic kidney disease at an earlier stage than we can at the moment. So quite a few potentials for it, but at early stages at the moment.
So these things that I'm taking out here are my screen printed disposable electrodes, and basically what they're in at the minute is a solution of phosphorus pentachloride and ether, which is why it doesn't smell very nice, and they're essentially modifying the surface so that I can attach DNA to the surface. In kidney disease and urine you have different levels of the RNA, and then if we can quantify the RNA we can tell you if you've got kidney disease or not. So we're trying to develop these electrodes as first means tests that can be done at the GP. So you see how a voltage is applied, as voltage applies, a current is read until it reaches a peak maximum, and then that's where the electrolyte is oxidized, and then it will come back the other way and apply a negative voltage which will cause it to reduce. And then the bigger these peaks are, the more RNA is on my surface. I mean, what I just did there would be what you do in the doctor's office.
So I collect and analyse all data for all whales on the acute kidney injury electronic alerts, and then on a quarterly basis then health boards can get reports on their incidents, their outcomes of AKI. The data also supports quality improvement projects throughout the health board across all different hospitals in Wales.
This is the tissue bank. So we are in the freezing facility for the Wales Kidney Research Tissue Bank, which is a collection of samples donated by patients and healthy volunteers. So we have a range of various type of samples which are ready to be transferred to researchers when they apply to the tissue bank.
Okay, so I'm taking samples out of the centrifuge. Basically what I've done is, these are cells from kidneys, and what I've done is I've put them in triazole, I've dissolved all the cell components, and the RNA, I don't know whether you can see the top. So the RNA is the clear liquid at the top, and that's what I'm going to take off the sample. And I'm going to leave the pink stuff because that's all the DNA and all the other cellular stuff. So basically it just involves trying to be really careful and taking off the top layer and leaving the middle layer and the bottom layer. So I'm just taking that layer off and putting it into a new tube. So that layer is the RNA, and after that I'm going to purify the RNA a little bit more, and then that'll go on for analysis to see what microRNAs are in there. So like I said, these are from kidney cells that I've grown up, and I've treated them with glucose and TNF alpha, so they're both things that you'd find in a sort of diabetic kidney. So I'm going to see how that influences the microRNAs that they send out, basically.